Hello and welcome back. So in this video we will see regarding citric acid cycle, or also known as TCA cycle, or also known as Krebs cycle. So before starting with the TCA cycle, we will have a brief introduction regarding glycolysis and PDH complex. Already this glycolysis and PDH complexes have been defined in two different videos that have already been uploaded. So you can go through those videos. But now just we will brief out. So in case of glycolysis, glucose is being converted into pyruvate, and this reaction takes place in the cytosol of a cell. And from this conversion of glucose to pyruvate, 8 ATP molecules are being generated. And this glycolysis process takes place in 10 different steps, including 3 different phases of energy investment phase, splitting phase, and energy generation phase. Next, the pyruvate, whatever is formed because of the glycolysis, is transferred to mitochondrial membrane, that is inner membrane of the mitochondria, using different transport proteins. So this pyruvate is being oxidatively decarboxylated into acetyl-CoA using PDH complex, that is phosphate dehydrogenase complex, that involves 3 different enzymes and 5 different cofactors or coenzymes. So including 3 enzymes and 5 cofactors, this PDH complex is being generated. So this PDH complex involves conversion of pyruvate to acetyl-CoA, and it takes place in the inner membrane of mitochondria. This conversion to acetyl-CoA takes place only in aerobic conditions and results in generation of 3 molecules of ATP. So this acetyl-CoA, as it is present in the inner membrane of the mitochondria, from the inner membrane it is being transferred to the mitochondrial matrix. So in the mitochondrial matrix, the Krebs cycle is being carried out. So TCA cycle, also known as tricarboxylic acid cycle or Krebs cycle, or the citric acid cycle. So this involves oxidation of acetyl-CoA. From where does this acetyl-CoA comes from? From pyruvate dehydrogenase complex reaction, wherein pyruvate is being oxidatively decarboxylated to acetyl-CoA. Now in TCA cycle, again the acetyl-CoA is being oxidized to produce carbon dioxide and water. And also there is a release of energy in the form of NADH, FADH2 and GTP. So this NADH and FADH2, they further undergo electron transport chain, wherein ATP molecules are being synthesized. This is how oxidation of acetyl-CoA is being carried out using citric acid cycle. The final pathway where the oxidative metabolism of carbohydrates, amino acids and fatty acids converge, their carbon skeleton being converted into carbon dioxide. So this TCA cycle is also important, and it is a final metabolic pathway, or you can say final oxidative metabolic pathway of carbohydrates, also for amino acids and for fatty acids. It is not only taken only for the carbohydrates, it can be also taken place for amino acids and fatty acids, wherein the finally acetyl-CoA is only produced as a final oxidative product of different metabolic reactions, and this acetyl-CoA undergoes TCA cycle to produce carbon dioxide and water. So the enzymes of TCA cycle are present only in the mitochondrial matrix. So you have to remember each and every step, where does it takes place. Like glycolysis, it takes place in cytosol. PDH complex reactions takes place in the inner membrane of the mitochondria. This TCA cycle takes place in the mitochondrial matrix. And similarly for ETC also we will see that it takes place in the inner membrane of mitochondria itself. So this we will see in the next video regarding electron transport chain. So for now you have to remember that TCA cycle takes place in the mitochondrial matrix. Next, the important point to remember is oxaloacetate. This oxaloacetate is one of the important intermediary compound of TCA cycle, and this oxaloacetate plays a catalytic role in citric acid cycle. It plays a catalytic role in citric acid cycle. We will see how it plays catalytic role in citric acid cycle. So now let us see in detail how citric acid cycle is being carried out. So this is the pathway for citric acid cycle. You can see the very first step, that is glycolysis, wherein glucose is being converted to pyruvate. This takes place in the cytosol. Now this pyruvate is being oxidized to acetyl-CoA, and this takes place using pyruvate dehydrogenase complex in the inner mitochondrial matrix. Now from this inner mitochondrial matrix, acetyl-CoA is being transferred into the mitochondrial matrix, wherein this acetyl-CoA and oxaloacetate, I said now in the that oxaloacetate plays a catalytic role in citric acid cycle. Now this oxaloacetate reacts with acetyl-CoA in presence of enzyme citrate synthase and results in the synthesis of citrate. Enzyme responsible is citrate synthase because citrate is being synthesized, so the enzyme used is citrate synthase, wherein the acetyl group is being transferred to oxaloacetate. You can see this acetyl group here is being transferred to oxaloacetate to form citrate, and coenzyme A molecule is being lost. Coenzyme A molecule is being removed and only the acetyl group is being transferred to oxaloacetate. Now the second step involves isomerization of citrate. You can see here the hydroxyl groups and the hydrogen groups of the citrate are interchanged between the adjacent carbon atoms. At second position in the isocitrate there is only hydrogen, while in case of citrate there is hydroxy, and this hydroxy is isomerized to form at the first position. So there is generation of isocitrate. This citrate and isocitrate are isomers of each other. So there is isomerization reaction taking place in presence of enzyme aconitase. The third step involves conversion of isocitrate to alpha ketoglutarate. So this conversion, or also you can say there is an oxidation of isocitrate to alpha ketoglutarate. As you can see, the hydrogens are being removed and there is a formation of ketonic functionality. So alpha ketoglutarate. So isocitrate is oxidized to alpha ketoglutarate. So these hydrogen atoms or hydrogen molecule that is being removed is accepted by NAD+. So in all the cycles, this NAD plus is being reduced to NADH by accepting a pair of hydrogen. So these hydrogens are being transferred to NAD plus and NADH is reduced form is obtained, along with elimination of a carbon dioxide molecule to form alpha ketoglutarate. So enzyme responsible for conversion of isocitrate to alpha ketoglutarate is known as isocitrate dehydrogenase. The name itself indicates that there is a dehydrogenation reaction taking place from the substrate known as isocitrate. The next step involves conversion of alpha ketoglutarate to succinyl coenzyme A. So again here the coenzyme A molecule, in the very first step we had seen the coenzyme A molecule is being removed from acetyl-CoA. So this coenzyme A molecule now reacts with alpha ketoglutarate. It adds to alpha ketoglutarate along with elimination of a carbon dioxide molecule. So there is elimination of carbon dioxide and addition of coenzyme A molecule, resulting in formation of succinyl-CoA along with removal of hydrogen. So here three processes takes place. One is oxidation, that is removal of hydrogen. Another is decarboxylation, that is carbon dioxide molecule is being removed, and another is addition of coenzyme A molecules. So three different processes are being taking place for conversion of alpha ketoglutarate to succinyl-CoA. Whatever the hydrogens are being lost is being accepted by NAD+, and this NAD+ is reduced to NADH. So this reaction takes place in presence of enzyme alpha ketoglutarate dehydrogenase. So alpha ketoglutarate is a substrate that undergoes dehydrogenation reaction to form succinyl-CoA. Next step involves conversion of succinyl-CoA to succinate. Succinyl-CoA to succinate, wherein the coenzyme A molecule that was attached or that was added is being removed in presence of enzyme succinyl-CoA synthetase. Don't get confused between synthase and synthetase. Synthase is that synthesizes, while the synthetase enzyme here, the role of this synthetase enzyme is to remove the coenzyme A molecule from succinyl-CoA and form succinate. For this reaction, there is addition of the inorganic phosphate that causes conversion of GDP to GTP. means while the elimination of succinyl-CoA, energy is being released. So that energy is utilized for addition of this phosphate group to GDP, that is guanosine diphosphate. So this GDP accepts this phosphate, and the energy whatever is lost by the release of coenzyme A molecule is utilized for synthesis of GTP. Now this GTP is analogous to ATP. You know ATP, adenosine triphosphate, and guanosine triphosphate. So these both are analogous to each other. So there is conversion of succinyl-CoA to succinate along with synthesis of GTP. So now here we can assume that this GTP is analogous to ATP, thus energy is being synthesized during conversion of succinyl-CoA to succinate. Now this method wherein the substrate directly produces the energy is known as substrate level phosphorylation. This we have already learned in glycolytic pathway, wherein the different substrate in the energy production phase produce directly ATP. There is no need of electron transport chain to be undergone for synthesis of ATP. Similarly, here also, while conversion of succinyl-CoA to succinate, there is a synthesis of GTP, that is nothing but ATP. Hence, this process is known as substrate level phosphorylation. Now, this succinate enzyme undergoes conversion to fumarate. You can see there is oxidation reaction again. So the hydrogens are being lost and double bond is being formed. So undergoes oxidation to form fumarate. So these lost hydrogens are accepted by FAD, that is flavin adenine dinucleotide, and it gets reduced to FADH2. And the enzyme responsible for conversion of succinate to fumarate is known as succinate dehydrogenase. There is succinate dehydrogenase, that is hydrogen is being lost. Now this fumarate is converted into malate in presence of enzyme fumarase. You can see in enzyme fumarase, wherein a water molecule is being added and these unsaturated bonds are being broken down and saturated bond is formed along with addition of hydroxyl group. You can see there is addition of hydroxyl group, formation of single bond from double bond because of addition of water in presence of enzyme fumarase. Now the last step involves conversion of malate back to oxaloacetate. So this malate undergoes oxidation in presence of enzyme malate dehydrogenase. Wherever the enzyme dehydrogenase is being present, you can see a different malate dehydrogenase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and succinate dehydrogenase. So in these places, either NADH plus or FAD is being taken into account, and there is reduction of these coenzyme molecules. You can see there is a reduction of these coenzyme molecules and there is removal of hydrogen from the substrate, whatever it is involved. There is removal of hydrogen from the substrate. So dehydrogenase is responsible for removal of hydrogens from the substrate and reduction of the coenzymes. So here malate dehydrogenase enzyme converts malate to oxaloacetate. Now this oxaloacetate again it accepts another acetyl-CoA molecule and the cycle restarts. So this is how acetyl-CoA undergoes oxidation reactions to produce carbon dioxide and water molecule along with reduction of different coenzymes to NADH and FADH2 respectively. So these coenzymes reduced undergo electron transport chain to produce different amount of ATPs. So here the role of oxaloacetate is to accept the acetyl group and convert it into citrate. If there is deficiency of this oxaloacetate or if there is decreased amount of oxaloacetate in the mitochondrial matrix, then there is no reaction with the acetyl-CoA and there is no formation of citrate. So the whole cycle will be incomplete if this oxaloacetate molecule is not replenished or if the oxaloacetate concentration is reduced in the mitochondrial matrix. Hence, oxaloacetate plays a catalytic role in the citric acid cycle. This is the reason why oxaloacetate is important substrate in the citric acid cycle, or you can say important intermediate in the citric acid cycle. Now let us see the energetics. So here you can see there is one NAD plus being converted to NADH while conversion of isocitrate to alpha ketoglutarate. The second is from alpha ketoglutarate again another NAD plus is reduced to NADH. Third NAD plus molecule from malate. So totally 3 NAD plus being reduced to NADH from malate. One FADH2 is produced from succinate and one is substrate level phosphorylation, that is synthesis of GTP from succinyl CoA. As we have already learned that each molecule of NADH produces 3 molecules of ATP and each molecule of FADH2 produces 2 molecules of ATP. Thus, the ATP produced is 12 molecules totally, that is 3 plus 3 plus 3, that is because of 3 NADH, that will be multiplied by 3, that will be 9 ATPs and 2 FADH2. 2 FADH2 from succinate. 9 plus 2 will be 11 and 1 is GTP, that is analogous to ATP, so we will consider it as only 1. So totally 12 molecules of ATP are being produced while no ATP is being consumed. We have not seen any of the ATP being consumed, only ATP is being synthesized. So in case of TCA cycle, no ATP is being consumed, only ATP is being synthesized from different coenzymes and different analogous molecules like GTP. So total ATP production from TCA cycle will be 12 ATPs. This is the maximum number of ATPs being synthesized during cellular oxidations. So now how this TCA cycle is being regulated. The very first enzyme, that is citrate synthase. You can see in this first citrate synthase, right? That is conversion or synthesis of citrate from oxaloacetate and acetyl CoA. So now this citrate synthase is inhibited by ATP and NADH because if there is sufficient amount of ATP present in the body and if this NADH molecules are sufficient to produce ATP from electron transport chain, this citrate synthase must be inhibited so that further ATP synthesis is being stopped. So the citrate synthase is inhibited by ATP as well as NADH. The isocitrate dehydrogenase, the enzyme that is responsible for synthesis of alpha ketoglutarate, right? That is the enzyme. This enzyme is activated by ADP because ADP, as we know, ADP is adenosine diphosphate. So there is deficiency of energy in the body. In such case, these ADP molecules are high. So these ADP will activate isocitrate dehydrogenase that will in turn convert alpha ketoglutarate. Here you can see this isocitrate is being converted into alpha ketoglutarate in presence of enzyme isocitrate dehydrogenase. So this enzyme is activated by ADP so that ATP synthesis is being increased, and also this is inhibited by ATP and NADH. Next is alpha ketoglutarate dehydrogenase. This is inhibited by succinyl CoA and NADH. So alpha ketoglutarate dehydrogenase, this enzyme is being inhibited by its product itself. You can see the product itself is succinyl coenzyme A and NADH. These two are the products that inhibits itself enzyme alpha ketoglutarate dehydrogenase. Next is the concentration of ADP. It is very important because citric acid cycle can be proceeded or citric acid cycle can be carried out if ADP concentration is more, that is energy is less, right? If ADP is more, that means ATP is less. So in order to replenish the ATP levels or the energy levels must be increased, this ADP activates the citric acid cycle. Next, accumulation of NADH and FADH2 will lead to inhibition of the enzymes and also limits the supply of NAD plus and FAD. So as we know, these NADH and FADH2 will undergo ETC and will produce ATP. If there is more and more amount of NADH and FADH2 produced from acetyl CoA, then there will be accumulation of NADH and FADH2, and these NADH and FADH2 molecules will undergo ETC and there will be enormous amount of energy generated. So in order to inhibit that is in order to prevent excess of energy generated. These NADH and FADH2 inhibit the enzymes that are responsible for TCA cycle to be carried out. Hence, the levels of NADH, FADH2 and ADP is very important in order to maintain or in order to regulate the TCA cycle. The important point regarding the TCA cycle is its amphibolic nature. What do you mean by amphibolic in nature? You know, amphibians, those are the creatures that live in land as well as water. That is, they have two characteristics. Similarly, citric acid cycle also has two characteristics. One is catabolic characteristic, another is anabolic characteristic. What do you mean by catabolic characteristics? Catabolism is nothing but conversion of complex molecules into simpler forms and along with the generation of energy. And anabolic process is conversion of simpler forms into complex forms with the utilization of energy. Right. So hence catabolic nature, we had already seen that the acetyl CoA is a complex molecule that is being converted into carbon dioxide and water. So complex molecules being converted into simpler forms along with generation of energy in the form of NADH and FADH2. Next, what do you mean by anabolic nature of the citric acid cycle? Anabolic nature includes conversion of oxaloacetate or alpha ketoglutarate. This is not O, this is alpha ketoglutarate into non-essential amino acids like aspartic acid and glutamic acid. Now, in case of anabolic reactions, these oxaloacetate and alpha ketoglutarate acts as a simpler molecules. Even though they are complex in nature, but here they act as a simpler molecules and result in generation of complex molecules like aspartic acid and glutamic acid. These two, aspartic acid and glutamic acids, are different types of non-essential amino acids. We had seen in the topic biomolecules non-essential amino acids, that is aspartic acid and glutamic acid. So from oxaloacetate and alpha ketoglutarate, that are intermediates of TCA cycle, anabolic reactions are being carried out that result in synthesis of aspartic acid and glutamic acid. The another important intermediate, that is succinyl CoA, is used for the synthesis of heme. So this heme is very important and essential component of electron transport chain. So this electron transport chain consists of different complexes. So one of the complex includes the heme as its internal part, or you can say as internal protein. So we will see this, how this heme is responsible for complex formation in the ETC and how it is utilized for synthesis of ATP in our next video, that is electron transport chain. Next is mitochondrial citrate. So this citrate, you had seen citric acid. So this citric acid present in the mitochondrial matrix is being transported into the cytosol. It is transported to cytosol where it is degraded into acetyl CoA. You can see citrate again being degraded into acetyl CoA only when it is transferred to the cytosol. So this acetyl CoA is utilized for synthesis of fatty acids and sterols. So these fatty acids and sterols are responsible for synthesis of lipids. So indirectly, the citric acid cycle, anabolic reactions of citric acid cycle are responsible for synthesis of macromolecules like proteins from non-essential amino acids and lipids from sterols and fatty acids. This is how amphibolic nature of citric acid cycle is being defined. That is catabolic reactions, that is citric acid cycle itself, and the anabolic reactions like conversion of oxaloacetate and alpha ketoglutarate into non-essential amino acids and mitochondrial citrate into fatty acids and sterols. So next we will see what do you mean by anaplerotic reactions. So the reactions that are concerned to replenish or fill up the intermediates of the citric acid cycle. Now the intermediates involve oxaloacetate, citrate, alpha ketoglutarate, succinyl CoA and malate. So these are the important intermediates that must be replenished time by time so that citric acid cycle takes place without any disturbance. So whatever reactions that replenishes these important intermediates of TCA cycle are known as anaplerotic reaction, or the reaction is also known as anaplerosis. You can see here alpha ketoglutarate being undergoes anabolic reactions to form glutamate, and this glutamate undergoes different series of reactions to produce non-essential amino acids like glutamic acid. So this is anabolic reaction. So now if this non-essential amino acids get degraded and it produces alpha ketoglutarate, right? If these non-essential amino acids degrade and they produce alpha ketoglutarate, this is known as catabolic reaction because the complex molecules produce the simpler forms. Now such reactions wherein the complex molecules are being produced to simpler forms, and those simpler forms are intermediates of the TCA cycle, then such reactions are known as anaplerotic reactions. So this is regarding the anaplerotic reactions wherein the intermediates of the citric acid cycle must be continuously replenished in order to effectively carry out the citric acid cycle, that is in order to effectively carry out oxidation of acetyl CoA. So now let us see what is the significance of this citric acid cycle. So as we have learned, catabolic reactions and anabolic reactions of citric acid cycle. So catabolic reactions are important for oxidation of acetyl CoA and also to synthesize energy in the form of NADH and FADH2, right? So one of the important significance is oxidation of acetyl CoA. Second significance is synthesis of NAD and FADH2, which undergoes ETC to produce ATP. ETC is nothing but electron transport chain to produce ATP. Another significance of TCA cycle is anabolic reactions, such that there is synthesis of macromolecules like amino acids, purines, pyrimidines, heme, fatty acids and sterols. So all these biomolecules are responsible for synthesis of macromolecules like proteins, nucleic acids like DNA and RNA, and also the lipids. So this is the significance wherein it produces energy as well as the simpler molecules like carbon dioxide and water. Also this TCA cycle helps in the synthesis of complex molecules like amino acids and fatty acids and sterols. So in this video we had learned the detailed procedure how citric acid cycle is being carried out, that is how the acetyl CoA is being converted into carbon dioxide and water, and also how the anabolic reactions are being taken place for synthesis of complex molecules. So in the next video we will see easy method to remember different intermediates of the citric acid cycle, and also how to remember different enzymes involved in the citric acid cycle. Thank you.